Hello, it's really my great pleasure to introduce today's speaker, Peng Yang. Peng did his postdoc at Dana Farber, and so I know him very well from our collaboration during that time. Peng did his PhD in computer science at Princeton University, then joined Shirley Liu's group at Dana Farber, and he's now an independent investigator at NCI. So Peng has a dual passion for both computational biology and immunology. And what I've always impressed about Peng is that he doesn't not only understand the computation deeply, but he also has a deep understanding of the biology, the relevant pathways, and an interest in actually using computation to advance the development of cancer immunotherapies. So an interesting example is a great paper he published in 2018 in Nature Medicine, where he reported a new computational platform called TIDE to identify genes that contribute to T-cell dysfunction and exclusion in tumors. And since he's been an independent investigator, he has taken up an interesting new line of investigation where he systematically studies cytokine signaling activity in tissues and at the single cell level. He had an elegant paper in Nature Methods in 2021. And now he's using that platform to study immune cell function in tumors. He has a really great paper on T-cell resilience in immunotherapy that was recently published in Nature Medicine. And in addition to all of these amazing talents, he also has a talent in music. So he had a Carnegie Hall recitation. He won the first prize for the Princeton International Chinese Music Festival competition when he was in college. And so Peng, it's really a great pleasure to have you here, and please deliver your talk.
Okay, Kai, thank you so much for the fantastic introduction. And also thank you so much for hosting me today. Today is really my great honor to give this presentation to Dana Farber, the place where I grew up, for my true scientific enthusiasm and scientific niche. So as Kai introduced, my background is originally from the computer science side, and most people know me for the work of TIDE, which is a data science framework to evaluate biomarkers and the regulators of cancer immunotherapy. And currently, my research group focuses on developing big data frameworks for cancer immunologists to make discoveries through reusing the large amount of public data. In other words, my job is to make sure that the immuno-oncologists here and cancer immunologists here, you guys can make the discoveries through the data platform we provide. And today, I would like to introduce two recent frameworks that we have developed. The first one is called the CytoSig database for analyzing cytokine signaling cascade and predicting cytokine target activities using gene expression data. And the second one is called the TRAS framework for analyzing marker genes of T cells that are resilient in solid tumors and predicting anti-tumor efficacy of T cells by their expression profiles. So the first work is a foundation of the second work. Is in collaboration with Kai. Cytokine is the basic signal that the immunology system utilizes to relay message. And the misregulation of cytokine activities could cause many types of human inflammatory disease. So also the proper anti-tumor immune response is regulated by many cytokine signals, which guide T cells into the tumor microenvironment to exert proper functions. Currently, our basic tool to study cytokines are ELISA and LUMINEX assays to measure the cytokine release. However, the cytokine release is very transient. If you don't measure it at the right time point, you are going to miss the signal. And also a cytokine level in a sample is not equal to its function activities in recipient cells. Also, cytokine functions have two vaccine properties, which are their complexity and redundancy. So to address this challenge, we have developed a computational framework to answer two questions for immunologists. First question is, which cytokines are regulating my genes of focus? Second question is, what are the cytokine signaling activities in your sample? So the name of our framework is CytoSig, representing cytokine signaling analyzer. And the cytokine has two usages. The first usage, you can use CytoSig as a database of cytokine regulatory relationships. You can query a gene name to view the upstream cytokine regulators or downstream target genes if your query is a cytokine. In the second usage, you can predict signaling activities for about 43 cytokines through an input sample's expression profile. And this work is in collaboration with Kai. To build a database, of course, the first thing is always the data collection. So there is a large number of cytokine treatment response data available in databases such as NCBI GEO and ArrayExpress. We hypothesized that this public data could serve as a knowledge base to model signaling activities in diverse biological contexts. However, the experimental design behind each publication is unique, requiring very labor-intensive expert interpretation and the standardization of the metadata. To overcome these hurdles, we established the framework for data curation. It's here, which couples large-scale automatic data processing of RNA-seq and microarray data with some natural language processing functions to enable systematic data curation. Finally, with this framework, we have labeled more than 20,000 cytokine treatment response profiles, and each profiles have their own biological replicates. So you can see there is a strong study bias. Some cytokines, such as TGI-beta and interferons, have lots of data generated, while most other human cytokines are poorly covered in published studies. By the way, we group cytokine, chemokine, and growth factors all together and use cytokine as a unified name for the sake of simplicity. So there are about 200 cytokines in human according to a textbook estimation. And for most of them, unfortunately, we don't have any treatment response profiles available for now. So here are some example data from our curation. Each dot here represents one profile generated from some cell models with biological replicates merged. The y-axis is the treatment cytokine, and the x-axis is a target gene. Our data collection will capture known knowledge of cytokine pro-inflammatory cascade. For example, IL-12 induce interferon, and interferon induce CXL 9, 10, 11 to attract lymphocyte. And you can see that their response profile all have the positive log-fold change in our curated data. When we focused on the regulations among cytokines, we found two major clusters. The first cluster comprises interferons as regulators and a few pro-inflammatory signals as target. And the second group comprises a few cytokines that signals through NF-kappa B pathway as regulators and a few pro-inflammatory signals distinct from the first group as a unique target. I have to say that the current bicluster structure is potentially due to the current data collection bias from the literature. And once we have a more cytokine treatment response data set available, maybe the bicluster structure will change. An essential question is whether our data collection represents the cytokine regulation in human physiological relevant settings. Because these data are largely generated in cell models, which may not really represent the real situation in human tissues. Therefore, we analyze the correlation between cytokines and their target genes captured in each data set. So in this example, we show it from a IL-10 treatment response experiment from a monocyte model. The x-axis represents the IL-10 ligand or receptor gene expression in TCGA lung cancer data. And the y-axis represents a target score representing the average gene expression of the target captured in this monocyte treatment response data. You can see that pretty much you see a perfect positive correlation between IL-10 expression and the target score in this bulk tumor tissue example data set. This positive correlation is true for most data sets from TCGA and GTEx human tissue data, covering thousands of samples from human cancer and normal tissue types. So this is showing that the cytokine response data we capture from cell models are largely relevant in the human physiological relevant tissue settings. So encouraged by the high quality, we analyzed the regulatory patterns revealed by our data collection. For example, you can see that IL-1 beta and TNF alpha tends to co-regulate many target genes. This is consistent with their known functions commonly through NF-kappa B signaling. However, we also observe that cytokines may fight against each other through antagonizing their downstream target genes in about 14% pairs. For example, our data indicate that the BMP6 may antagonize the effect of IL-1 beta through downregulating IL-1 beta-induced targets, such as CXL1, 8, and CCL2. To the best of our knowledge, BMP6, a growth factor, is not known as an anti-inflammatory signal that antagonizes the IL-1 beta signaling activity. So we perform flow analysis of intracellular protein level and ELISA assay of secretion level on some human epithelial cell lines. This result indicates that the level of CXL8, CCL2 on protein level are consistently lower in cells treated with BMP6 plus IL-1 beta compared to IL-1 beta treatment alone. So the data collection also enabled us to predict cytokine signaling activities using an input sample's gene expression profile. So we utilize a very simple linear regression model to decompose cytokine response patterns from an input sample's expression profile. For example, upon IL-1 beta neutralizing therapy among a thoracic patient, the IL-1 beta activity reduction at day 3 predicted by cytosig on the y-axis correlates significantly with the patient clinical response at day 15 on the x-axis. So which means that the patient response to the IL-1 beta neutralizing therapy is already evident on day 3 if you use the cytosig as a biomarker. As another example, in the interferon alpha vaccine trial among seismic lupus patients, the interferon activity reduction predicted by cytosig using the peripheral blood expression profiles correlate significantly with the clinical response measured by IL-1 alpha neutralizing antibody titers. And there are some further examples from anti-VEGF therapy studies. So we predict the VEGF signaling activities using the pre-treatment expression profiles of patient tumors. For example, this is the clinical trial, BioLab phase 2 clinical trial in GBM patient. The pre-treatment activity of VEGF signaling is quite predictive of anti-VEGF survival outcome in 2 independent clinical studies, which is as expected. And there are some clinical trials showing that anti-VEGF does not really bring additional clinical benefits in the GBM patient. compared to conventional therapies. And I think our algorithm demonstrates the potential opportunity to rescue those failed clinical trials because they really need to focus on the patient with the existing pre-treatment VEGF signaling activities. Otherwise, the antibody wouldn't work. So we comprehensively collected transcriptomic profiles from anti-cytokine therapy clinical studies or mouse models if clinical data is not available. The cytosig prediction generally agree well with the clinical outcome in about 80%, 85%. In other words, if the cytosig predict a reduction of cytokine activity using a patient sample, that patient typically will respond to the relevant anti-cytokine treatment. A surprising result is that cytosig predicted different activities for TGF-beta 1 and 3, which all share the same receptors. Initially, we are skeptical that whether these two isoforms have a true distinct activity because they share the same receptors. To validate our computational prediction, we utilize the different antibodies that neutralize different isoforms of TGF-beta to treat the 41 tumor models. So in this case, we can create a differential profiles for pan-TGF-beta inhibition, 1,2 specific inhibition, and also a TGF-beta 3 specific inhibition. And the cytosig prediction of the isoform specific activity in general agree pretty well with the neutralizing antibody treatment differential expression profiles, indicating indeed the isoform specific prediction of cytosig is statistically reliable. So we also evaluate the prediction performance of cytosig using single cell RNA-seq data. Our gold standard is the transcription factor activity predicted using ChIP-seq dataset and my previous software Rabbit. And we found that the cytokine activity predicted is typically very consistent with the predicted activity of their known downstream transcription factors. So for most cytokine and transcription factor combinations, such consistent results are true across many single-cell RNA-seq dataset from diverse disease settings. This result indicates a good quality of cytosig predictions on single-cell data, at least measured by the transcription factor signaling activities on the downstream of cytokine. So finally, we applied cytosig to study some molecular features of patients with severe COVID-19 symptoms compared to mild controls. We utilized the single-cell data published for patient lavage and peripheral blood samples and search for cytokine activities that are significantly different between severe and mild patients. For example, IL-10 activities are significantly higher among macrophage from patient lavage samples in the severe patient compared to mild controls, and the type 1 interferon activities are only high among CD T cells from patients with mild symptoms. And overall, we found a group of signature cytokines for severe patients, such as IL-10, IL-1 beta activity are high, and in contrast, BMP6 activity are low for the severe patient. And the cytosig can indicate a regulatory network for these signature cytokines. For example, we found that the activated cytokines tends to co-regulate a few common target genes such as CXL8 and CXL1. Moreover, these chemokines are also target genes repressed by the unactivated cytokines in the severe patient. And indeed, their target expression level are significantly higher among severe patients, which is as expected. Currently, there are some clinical trials of blocking CXL8 or CXCR2 ligands as anti-cancer therapies, some of which already passed the phase 1 toxicity test. This result indicates the potential of repurposing these candidate anti-cancer drugs as COVID-19 relieving therapies. As a summary, we have developed a computational framework to query regulators or target in cytokine regulatory cascade. The cytokine can reliably predict the cytokine signaling activity using both bulk and single cell transcriptomic levels, and our data collection revealed the new knowledge such as BMP6 as an anti-inflammatory signal and the potential value of anti-CXCR2 ligands as potential relieving therapies for severe COVID-19 symptoms. So welcome to play with our online framework, and all of our source code are available also. So next, I will introduce another framework we developed recently called TRAS, which was developed based on cytosig to specifically help the field of T-cell-based immunotherapies. Here is our motivation. Cancer immunotherapy based on T-cells has led to significant success in some fraction of patients. However, most patients with solid tumors do not respond to these immunotherapies. And CAR-T therapy is only effective in blood cancers, but not solid tumors. The key reason for, or at least one of the key reasons for this gap is that solid tumor is a very hostile environment for T-cell to work. When T-cells are trying to attack tumor will induce many immunosuppressive factors to strike back at the T-cells. So to overcome these limitations, we aim to develop a computational model to address two questions. The first question is, what T-cells are more resilient in the hostile tumor microenvironment? The second question is, should a patient take the financial and healthy risk of cellular immunotherapies? Because cell therapy is very expensive, especially in some countries where the insurance does not reimburse such an expensive therapy. And also it could induce very severe toxicity. So you need to make sure whether the T-cell product will work or somehow work before initiating the whole expensive process. So we developed a computational framework named tumor resilient T-cells, or TRAS. The TRAS model can predict the clinical efficacy of the T-cell product using the pre-treatment expression profiles or even pre-manufactured expression profiles I will demonstrate later. Also, the TRAS framework integrates lots of public data sets. It provides you a platform for evaluating T-cell biomarkers for immunotherapy response and prioritizing regulators and candidate therapeutic targets for T-cell anti-tumor efficacy. Further, the TRAS model revealed FIBP as a negative marker of T-cell anti-tumor efficacy and a potential new therapeutic target for solid tumors. So let's have a look at the TRAS model. Previously, we have developed a cytosig platform which can predict cytokine signaling activities in a single-cell transcriptomic dataset. In this example, using single-cell RNA-seq data from a melanoma tumor as example, the TGF-beta signaling activities in CD8 T cells computed by cytosig negatively correlate with the T cell proliferation levels inferred by the expression of cell cycle and DNA replication genes. However, this negative correlation is not always true. If we split T cells by the expression of a third gene, you will observe some different associations. For example, the negative correlation between immune suppression and T cell proliferation is only evident for T cells with either a low IL-7 receptor expression or with high FIBP expression levels. This observation shows the basic idea of TRAS model, searching for genes whose status can modulate the correlations between immunosuppression and T cell proliferation. The TRAS model uses two stages to identify gene markers of CD8 T cells that are resilient in solid tumors. The first stage quantifies the level of immunosuppression imposed on CD8 T cells in a single-cell RNA-seq dataset as the signaling activity of some immunosuppressive cytokines predicted by cytosec, such as TGF-beta, PGE2, and Treg triad. The second stage utilizes a variable interaction test to identify gene expression markers of T cells that are resilient to pro-inhibitive signals. The relationship between immunosuppression, T cell proliferation, and the expression status of a third gene can be evaluated through the interaction covariant in the linear regression. But forget about this model intuitively. TRAS is searching for expression markers of T cells that do not change their proliferation status under the immunosuppression in solid tumors. So still using the previous example from a melanoma tumor, we can compute one TRAS score for each gene as a dot here. The x-axis presents the T cell persistence signature score from a previous study, finding the expression signatures of tumor-infiltrating lymphocytes that can persist in solid tumors and induce a complete cancer regression in their model. And the y-axis is a TRAS score computed from our model. As you can see that the TRAS score are very consistent with previous T cell persistence signature in this tumor example. Also, this consistency is true across 168 tumors in 19 other cancer types from our single cell data collection. We further evaluate whether the TRAS model can predict anti-tumor efficacy of T cell in immunotherapy. Here's our prediction procedure. We first collect some training data, single cell RNA-seq from 168 tumors and 19 cancer types. For each tumor, we can compute one vector of a TRAS gene score. Then we get the median gene score across all tumors as one overall TRAS signature. For each T cell's bulk expression profile from our testing data, please note our testing data has no overlap with our training data. In this testing data, they provide you the bulk expression profiles of T cells, either isolated from the pre-treatment tumors for immune checkpoint blockade inhibitors, or infusion product for CAR T therapies, or the pre-manufactured samples for CAR T or TIL transfer therapies. Pre-manufactured samples means the sample you use to make the infusion product from the patient samples. And as a prediction, we use the Pearson correlation between the bulk T cell profiles and the one overall TRAS signature. If the correlation is positive, we predict the T cell sample as effective. Otherwise, we predict the T cell sample as non-effective. Here are some examples of the TRAS prediction. Each panel represents one study that generated gene expression of T cells from pre-treatment tumors. Please note that some of these studies are single cell studies, so we got the average expression of all T cells in each tumor as a pseudo bulk profile. Each dot represents one patient. Please note that all of the y-axis is a TRAS score correlation from our model, and you can see that the color indicates the actual clinical response measured in each study. In all data sets, the positive TRAS score from pre-treatment samples or infusion product indeed predicted the responder status. And there are many published T cell signatures that generate much clinical interest, such as the persistence signature from Rosenberg lab discussed before, and the central memory signatures from Dr. Rastifo that lots of people are using, and also a bunch of signatures from other people's study. And TRAS achieved a higher prediction accuracy across different clinical cohorts than all previous signature compared. We also evaluate TRAS model prediction using bulk RNA-seq profiles from pre-manufactured samples during cell therapies, such as tumors before TIL expansion, or blood lymphocyte before CAR-T lentiviral transduction. You can see that a positive Treg score correlation indicates a significant better overall survival outcome, or like a progression-free survival outcome after cell therapies. Also, compared to previous T-cell efficacy signatures, Tregs achieved the best prediction performance. And together, by the way, you can see that an interesting phenomenon is this central memory signature worked really well for the immune checkpoint blockade inhibitor settings. But for cellular therapy settings, it does not really work well. And the Tregs is really robust in different comparisons. Such advantage is particularly important for cell therapies, which is extremely expensive and potentially very toxic. If you can do the prediction or prioritization of the patient before even start the lentiviral infection, that will give a significant advantage. So encouraged by the clinical prediction performance, we further evaluate whether the Tregs model can reveal some therapeutic target to enhance T-cell immunotherapies. We prioritize the genes whose expression level are consistently associated with low Treg score and poor immunotherapy outcome in solid tumors. Our analysis only focuses on negative markers because therapeutic approaches, whether by compound, antibodies, or genome editing, primarily inhibit targeted genes. Among all human genes, FIBP ranked first as a negative marker. To the best of our knowledge, very little literature exists for FIBP, except that FIBP bind FGF1, which is how you get the name FGF1 interaction binding protein. And also, like FIBP in tumor cells may enhance chemotherapy resistance in some tests with some colorectal cancer cell line studies. The Treg score of FIBP are consistently negative across most cohort in our training data set. The FIBP expression are significantly lower in T cells from anti-PD-1 responder pre-treatment tumors than values from non-responder tumors. And among tumor samples obtained before expanding TILs for adoptive transfer, you can see that a high FIBP expression level indicate a worse overall survival outcome after TIL therapy. Also, high FIBP expression in bulk tumors correlates a T-cell dysfunction phenotype according to our previous TIDE model. Since FIBP is such an outstanding marker in our integrated analysis, we decided to test whether it is a regulator and potential therapeutic target. Tregs and other signatures analyzed above are designed to reveal marker genes of T-cell functions. However, gene markers only reflect associations, which may arise from indirect effect rather than causality. Establishing the mechanism of regulatory causality is desirable for the development of therapeutic applications. So we first utilize some in vitro co-cultures between TCR T cells from human and mouse donors and cancer cell lines with the paired antigens. The readout is cell growth and cytokine release from cancer cells and T cells, respectively. In this example, human T cells targeting NY-ESO1 antigen were incubated with A375 cells labeled with red fluorescent color. FIBP knockout T cells presented a much better killing efficacy than control AVS1 knockout T cells. We have evaluated the killing efficacy on T cells engineered from 2 different donors and found the result is quite consistent. The overall effect of FIBP knockout is similar to CBL-B knockout, which is a well-known control gene modification in T cells to enhance anti-tumor efficacy. And the CBL-B knockout is under clinical trial right now. So also FIBP knockout significantly enhanced the release of interferon gamma and TNF cytokines in both human and mouse donor T cells. Next, we evaluate the in vivo efficacy of FIBP knockout T cells by adaptive transfer in mice and evaluate the B16 tumor growth. We can see that the tumor size in mice transferred with FIBP knockout T cells is significantly smaller than tumors in mice transferred with controlled ROTA26 knockout T cells. And also, both FIBP and CBP knockout T cells promote a longer survival outcome. However, I have to say that the difference of our mouse growth curve is not really dramatic. We do double-blind measurement here, and also we do very rigorous control to make sure that all T cells just undergo the same set of genetic manipulations, and we can only achieve some statistical performance, but never this dramatic separation of the tumors here. To find the mechanism on why FIBP knockout can enhance the T cell anti-tumor efficacy, we perform some RNA-seq experiment in the mouse T cells with FIBP or ROTA26 knockout. This is a pathway enrichment analysis result on differential expression profiles between FIBP knockout compared to ROTA26 knockout. Surprisingly, all top results are related to the downregulation of cholesterol metabolism. For example, several genes encoding the enzyme of cholesterol biosynthesis, such as HMGCS, SQLE, are significantly downregulated. LDLR, low-density lipoprotein receptors, which transport fat molecules around the body, is also downregulated. In contrast, ABCE1, which pumps cholesterol out of the cells, is upregulated. So we validate the RNA-seq result using flow cytometry analysis on T cell cholesterol levels. Indeed, the cholesterol levels in T cells isolated from mouse tumors are significantly downregulated upon FIBP knockout before the adoptive transfer, but not ROTA26 or CBP knockout. Also, the cholesterol level are upregulated upon FIBP overexpression. In human bodies, the low-density lipoprotein are the major carriers of cholesterol across the body. And upon knockout of FIBP in T cells, the LDL absorption is significantly reduced. From the previous literature, cholesterol is known to inhibit effector T cell functions upon chronic exposure. Although cholesterol is required for T cell activation, but if you expose it for a long time, T cell will become dysfunction. Indeed, culturing T cells with ROTA26 control knockout in high cholesterol medium will significantly inhibit its tumor killing efficacy in the co-culture assay. However, T cells with FIBP knockout is quite resilient to high cholesterol levels in the medium, as tumor efficacy are killing efficacy are the same in normal and high cholesterol medium in this condition. So the FIBP knockout in T cells are making T cells more resilient to high cholesterol levels, which is a common feature of solid tumor microenvironment, because tumor cells, and many immunosuppressive cells, taking cholesterol as their food for malignant growth. Besides FIBP knockout, we also evaluate statin treatment on modulating the cholesterol level in T cells. As statin is a very common drug for lowering down cholesterol levels. Unsurprisingly, we only see a cholesterol lowering effect of statin treatment in T cell before T cell activation. However, after T cell activation, statin treatment even elevated the cholesterol content. This surprising result suggests that statin is not a really effective approach to control cholesterol levels in effector T cells. And by the way, a few other previous studies also report the cholesterol enhancing effect of statin in some cell lineage by a negative feedback loop to enhance the HMGCS expression level. So in summary, we developed a computational model named the tumor resilient T cells for studying T cell anti-tumor efficacy. Welcome to try our web framework, which integrates lots of public data sets to help you T cell study with these interactive function modules. Further, the TRAS model revealed FIBP as a negative marker of T-cell anti-tumor efficacy and potential therapeutic target. And last, like this work is done through our team effort. And in particular, I would deeply appreciate Kai for his continuing mentorship after my independence. Because at the beginning stage for a new faculty, I could make many mistakes. And Kai really helped like young faculty to avoid obvious mistakes through his experience and guidance. And also my colleagues and collaborators generously helped us by providing us equipment, mice, and even some precious in vivo data. And this is my team, and I really deeply enjoy working with them, and they really make this kind of work happen. And last, please allow me to somehow shamelessly give a little bit advertisement here. And we are recruiting right now for postdoc positions on computational biology, and also for doctoral students at the DFHCC community. You can even do thesis work at NIH under a partnership program, like if you are interested in working together. And thank you very much for your attention.
Thank you, Peng. That was really, really interesting. Mm-hmm. So let me see what questions we have in the Q&A. So the first question comes from Wubing Zhang, who you must know. So a great word, Peng, in your cytosig work, when you study the association between cytokine activity and anti-cytokine therapy response, do you only consider the activity of the target cytokine or other synergistic or anti-effect of other factors like BMP6, and how do you do that?
Yeah, I think this is a fantastic question, because the cytokine signaling activities will change. Like if the cell sends 2 or 3 cytokines together, and there are numerous examples of the combination of cytokine will create a different response on certain target genes. So in the cytosig, we only treat each cytokine treatment response profiles as one vector in the regression model, which means we only consider their linear combination without considering their combinatorial effect. The reason is the combination treatment data is very limited in the literature. And also, as you guys may observe, our prediction only aim to predict a weak conclusion, which is whether or not this cytokine exists, the signaling activity is activated or repressed in your sample, but we don't really predict the pathways or gene-specific behavior, because that could be largely affected by the combinatorial effect. However, if you're only interested in a yes or no answer on the activity level, then we observe that the single cytokine treatment response profile could work for most of the time. But indeed, because of the lack of data for combinatorial treatment, this is indeed a potential limitation for some cases.
Okay, great. And then Wubing has a second question. So in the second story, did you look at public CRISPR screen data on T cells and see overlap hits that are important for T cell activity?
Yes. Look at the public CRISPR screens on T cells. By the way, I want to mention that the public CRISPR screen on T cells from Alex Marcin's group on genome-wide has a very different endpoint. It's like an in vitro cytokine release and in vitro TCR, in vitro T cell growth upon TCR activation. And also before, Kai had some in vivo pool CRISPR screen data on a subset of potential regulators in T cells. So these are all very different endpoints. So I look at them and find a very low overlap. I need to be direct and honest on this. Because TRAS is more like a marker model, it's not a regulator model. So, and there are many studies showing the markers and the regulators do not really overlap with each other. And the FIBP, is only an outlier because its score is so extreme among so many markers. This is why we validate these most extreme ones. But for others, like I will say that the markers from T-RAS not necessarily means that that's a regulator. However, if anyone is developing some like a magnetic pull-down procedure to isolate T cells based on some cell surface markers, the T cell will be very useful. And also, if anyone is interested in predicting the patient response, then the biomarker model will be sufficient. But for genetic explorations, the T-RAS can only tell you whether your finding is clinically relevant, but it cannot replace the CRISPR model, which is still the direct measurement on genetic causality.
Great, thank you, Peng. So I have a question. So if you map your T-RAS signature onto T cell single cell data from human tumors, which T cell populations identified? Are these primarily memory-like cells, or
yes,
they're probably not effector cells?
Yeah. So like I try this, like largely this will be those set of quiescent memory-like cells. Like they tend to be more sturdy, and also like some cells that are more resilient to cholesterol intake. But of course, that also means they are metabolically more silent. So like broadly, those are correspond to those kind of quiescent, metabolically low activity like stem-like cells. Because anyhow, these markers are highly correlated with each other.
I see. And so the cells, so then the effector cells and also the rapidly proliferating cells are probably very sensitive to
yes. Yeah, exactly. Those things, they will be eliminated first.
Okay, so I think that explains the finding from the cellular therapy studies, because many studies, including Nicholas Stifo, have shown that memory populations are actually essential for the efficacy of cellular therapies.
Yeah.
And it's the memory populations that actually account for the in vivo persistence for or even.
But by the way, Kai, so like another interpretation is not really memory, it's the quiescence. Because if you see the sea turtle, they can survive in the deep ocean with very little food and low temperature. Because the sea turtle, they have very low metabolic activity to survive in a bad environment. Like an alternative interpretation of the memory studies is more about the T cell quiescence. Like for memory, it may or may not really be the marker. Maybe there are a secondary effect of T cell quiescence and low demanding. Like, what is your opinion toward this alternative interpretation?
Yeah, so I mean, quiescence and long-term survival. That's sort of the essence of the memory program.
Yeah.
But memory cells are different. Naive cells are also quiescent.
Yeah.
Memory cells obviously have the ability to do a rapid recall response within a few hours following re-challenge, right? And in that way, they're different from naive cells. But yes, but quiescence is sort of the essence of memory. I see, interesting.
Yes. And by the way, Kai, like I will say I do evaluate a bunch of previous markers, like so this marker, the central memory signature, is not really robust. It's really predictive in the checkpoint blockade settings, but for the pre-manufactured samples, this prediction performance is not very robust. This is why, for a long time, I'm wondering whether it's really quiescent. But you are right, at least they should have the ability to rapidly kill again after you re-challenge them.
Yeah, so I mean, when cells are manufactured in vitro, there are going to be very few cells that are truly quiescent, because during manufacturing, they all become activated. But some cells are more in a memory state, and some cells become effector cells, and some become, terminally differentiated effector cells. And so I think that, your signature identifies the cells that have more sort of long-term potential in vivo. So
yeah. And meanwhile, Kai, like some people criticize me that in general for this kind of strategy, it's very dangerous to make T cell really potent by any genetic modification, because CAR T and any kind of cellular therapy could already have some toxicity. You make them more sturdy, right? Like this means you could amplify their toxicity. Like this is some criticism I got when I talk about this work to AstraZeneca. So what is your opinion towards the potential negative effect of potentiating T cells?
Well, so I mean, if you encourage T cell, with therapeutic cells, you want persistence, right? I mean, if the cells are only around for a month, then they can't protect against, recurrence, months or years later. So I think you want persistence. What is dangerous is turning all of the cells into highly active effector cells that produce huge amounts of cytokines. And that gives you the toxicities associated with cytokine release syndrome and also the neurotoxicities. So I think, pushing the cells more into a memory state or persistent state is actually better in reducing toxicities. So
So
that's how I think about it.
I see. Good point. So in this case, you give patient less cells, but you make sure you give them less cells, but these cells are indeed in a good, sturdy state. And then you won't have a burst of cytokine at the very beginning, potentially. Yeah.
Cool.
Yeah.
Thank you so much. I don't think we have any further questions in the Q&A. So Peng, that was awesome. You did great. Yeah.
Yeah, thank you, Kai. Oh, by the way, Kai, before you leave, may I just continue a little bit of the discussion before our talk? Like you mentioned that you think the TCR therapy will be a very promising future directions, right? I'm just asking your opinion towards the TCR pool therapies. Do you think, because any single TCR therapies could lose its efficacy, do you think the TCR pool therapies will be the fundamental solution, or you think it's some alternative ones?
I think this is a longer discussion. So why don't we follow up? Because let's set up a time and maybe meet next week. And thank you for this amazing seminar.
Yeah, thank you so much, Kai, for hosting me, and thank